researchers. And another goal is to encourage discussion amongst an interdisciplinary community. So on that, the format for this talk will be two parts. We'll have two speakers today, and at the end of that, after they speak, we will hold a discussion. And so please hold any questions till the end. Okay, so for our speakers today, we will have Shu Chen speaking, and then Alberto Vazquez-Salazar speaking. And both of these researchers are from Southern California, Shu from UC San Diego and Alberto from UCLA. To give a topical introduction will be Professor Ulrich Muller from UC San Diego. And please go ahead and take the floor, Uli.
And I need to introduction. So since both Alberto and Shu will about audience diverse, I'm trying to introduce how their research fits into the broader community's efforts in study origins of life. So we're all interested in how life began. And we know that about 4.53 billion years ago, there was a solid crust on Earth. We know that about 3.5 billion years ago, there were living organisms, but we don't know how what came in between. And this is the focus of the research. And to think about that, I like to think about three different points that always need to be considered. One of them is we may all have different definitions of life, but I think it's very useful to have a working model of your definition of life so that you know what you're looking for. And I'm using the definition that life is a self-replicating molecular system that's capable of open-ended Darwinian evolution. So you need to have molecules that are able to self-replicate and evolve. And how exactly self-replication and evolution looks like may have been dramatically different for early stages. But by definition, because we are talking about the origin of today's life, there must have been some kind of continuity in terms of information transfer and metabolism. And what a lot of research is focused on is how could this be chemically realized? So we need to think about chemical pathways that both generate metabolites as well as the macromolecules that can then fold and catalyze those reactions. And in addition, I just see that my numbering is off because I resorted it this morning. In addition, we need to always satisfy the second law of thermodynamics. So since by definition, if we have self-replication of some kind of structure, the second law of thermodynamics tells us we need to couple such a self-replicating system to another entropy generating process. And on the next slides, I want to talk to you about different perspectives that can be used to think about how this could have played out in reality. And the first thing, which may have been the oldest perspective, is a biocentric perspective. We just think about how would the last universal common ancestor, or looker, how could that have looked like? And you may know this paper by Hagedal 2016, where they expanded the tree of life. And that shows looker may have been down here. And if you compare all these genomes, you can say looker must have had a ribosome that was able to encode peptide synthesis, used ATP, had some catalytic RNAs, was using amino acids and those things. But there's only so far you can go back. And the danger here is that if you use only the biocentric perspective, that you think teleological, which means You think after this step, there must have come this step, there must have come this step, but evolution does not plan ahead. So each step in evolution needs to have its own benefit. And the other thing that Ram Krishnamurti described nicely in his paper about the 18th camel is that many early strategies that may have been very important in early life forms may not exist anymore in today's strategies. So the biological perspective is very helpful in guiding us towards, for example, that there was a ribosome. RNA catalysts were important at some early stage, but it has its limitations. Another perspective is when we look at the earliest fossils, what are the earliest reliable fossils? And you may know the stromatolites in Shark Bay, Australia, and that are about 3.5 billion years old, where we can be very confident, 3.5 billion years old, there were life forms on this planet. However, these fossils, they are already highly evolved, and any macromolecules like DNA, RNA, protein were long degraded. And the other thing we need to be very careful about in those kind of studies is that if we look at microfossils, they are often very difficult to discern from abiogenic origins. And in addition to that perspective, I want to look at the perspective of geology and modeling geology. What can we say about the early Earth environment? And the surface temperature, I think the current consensus, or mostly consensus, is that we had relatively moderate conditions, moderate temperature due to isotope data on sediments and carbon dioxide cycling. For land surface area, you can still read anything between 5% and 100% or 120% of the current landmass for early life, for the origins of life. We had liquid water, monovalent cations were likely a bit higher. There was magnesium. Importantly, there was iron 2+ in the ocean that was there because we didn't have oxygen. And those are quite strong, reliable data. And we had a lot of carbon dioxide, which is also a quite reliable piece of information based on carbon dioxide cycling. Whether we had low or high hydrogen, methane, ammonia, is unclear because we may have had strongly reducing condition with with an impactor that had a solid metal core. So there are many different possibilities, and all we can do is we can say what is roughly plausible and work with that. Now, this here is a paper that I just want to mention. This is the paper that describes that we could have had strongly reducing conditions for about 100 million years. Now, a synthetic perspective to study origins of life has been very useful. And the first perspective there is to look at chemical pathways. The second one is on artificial catalysts. And the famous Miller-Urey spark experiment showed that you can generate amino acids relatively easy from water, ammonia, methane, carbon dioxide. And then when you look at the other molecules that are kind of necessary, must have originated somehow, nucleobases and nucleotides, those have been studied for several decades. And especially the Pauner lab and the Sutherland lab and Thomas Carell's lab and Sidney Becker's lab have made a lot of, gave a lot of insight in how nucleobases and nucleotides could have been generated. Now, you may have heard that the RNA world hypothesis may have been RNA, that today's protein, DNA, RNA organisms are very unlikely to have originated together, because it's likely that the protein that is encoded by the DNA together with the DNA that is replicated protein emerged at the same time. So Alex Rich, Carl Woese, Francis Crick, Leslie Orgel, they proposed that there was a stage in the early evolution of life in which RNA was the only genome encoded catalyst and provided the genome. And the strongest evidence that we have that this was indeed the case is the ribosome, which is the machinery in all cells that generates proteins, genetically encoded proteins. And this ribosome is a ribozyme, a catalytic RNA. Since this is a highly evolved ribozyme, that means there must have been many stages of evolution before the ribosome. So we can be very confident that before the last universal common ancestor that already had a ribosome, there was a stage where catalytic RNAs were important. And to study that, people need to generate catalytic RNAs in the lab, because most of those catalytic RNAs have been lost to history. And the way I just want to show the general principle of how those in vitro selection experiments to generate new ribozymes, how they work. Because both Chirhan's talk as well as Alberto's talk, they are based on in vitro selection experiments. So what you do is you first generate a random library that contains many randomized positions. For example, if you have 100 randomized positions, you have 4 to the power of 100 possibilities, or about 10 to the 60 sequences that are possible. We can never cover all those sequences because the Earth has only about 10 to the 50 atoms. But we can cover something like 10 to the 14 possibilities, and usually we can find catalytic sequences within them. So first we transcribe the DNA to RNA, and then this random library of RNA is incubated with some kind of substrate. And the trick for the really difficult part for each of these in vitro selection experiments is to isolate those RNAs that can catalyze a reaction and discard those that don't. And let's say we have 0.01% of RNAs that are now containing the catalysts, and we discard 99.99%, then we have a 10,000-fold enrichment for catalytic RNAs. The left part of the cycle is just to amplify these RNAs. And to do that, we need to reverse transcribe them, PCR amplify them, and transcribe them again. And now we have a library of RNAs that is enriched for those RNAs that can catalyze the reaction with the substrate. And by going multiple cycles through that selection, enrichment, amplification, selection, amplification, we can, after some rounds of this in vitro selection procedure, isolate catalytic RNAs and study them. And today's talks from Chirhan from my lab, as well as Alberto Vazquez-Salazar from Irene Chen's lab, they will talk about catalytic RNAs that have been developed by this in vitro selection process. And with these catalytic RNAs, we hope to be able to study important stages in early life forms that are, in Irene's case, based on nucleic acid oligomers that form catalysts. And I think Alberto is looking at full RNA catalysts, so that we hope in the long run we can generate self-replicating ribozymes and recapitulate the whole series of steps that must have been important for the origins of life. And with that, I'm done with my introduction and hope that you found it useful.
All right, Shu, please get your slides shared. All right, go ahead and take it away.
Okay, thank you. Thank you for the introduction, Uli. And I will be talking about my recent project about the in vitro selection of catalytic complexes composed of short nucleic acid fragments, as Uli just introduced. This project is in the context of an early stage of the RNA world, and we are trying to study at how they originate during the origination of the origin of RNA world. And there's a conundrum for the rise of an RNA world. On the one hand, longer RNA are more difficult to be synthesized in the prebiotic environment. And there are many model studies to polymerize activated mononucleotides into long RNA strands, and they were catalyzed by different catalysis and under different temperature conditions. The model studies, the products of these polymerization acids mostly generate short RNA oligomers. And in these two examples, with only uridine, the products goes up to around 14 nucleotides long, and with all 4 different nucleotides, the products generated from this assay was around 20 to 30 nucleotides long. So, but on the other hand, as we just introduced, we need RNA to catalyze. So, yeah, if we need RNA molecules in the early stage to catalyze important functions for the early evolution. And these ribozymes usually requires longer RNA sequences. For example, the GTP synthase ribozyme and the polymerase ribozyme, they are more than 100 and even more than 200 nucleotides long. So this is the question we are looking at, that we need long RNA sequences for functions, but we can mostly only produce short RNA oligomers in the prebiotically plausible environment. So the solution we propose to circumvent this conundrum is to form catalytic complexes by short oligonucleotides. And that is to show this idea before the long RNA strands can be synthesized prebiotically at an earlier stage where there were a pool of short RNA oligomers, they can already assemble with each other by base pairing and form the catalytic active structure. With this structure, the catalytic activity can already be achieved. This type of assembly and catalysis has been supported by some studies showing that the known ribozymes can be fragmented into multiple short pieces, short fragments, while still remaining their original activity. This hammerhead ribozyme can be fragmented into 2 strands, and they can catalyze the cleavage at this cleavage site. And a triphosphorylation ribozyme selected in our lab previously, it can be fragmented into 3 short fragments, and they can assembly and catalyze the 5' triphosphorylation of its own 5' hydroxyl group. So now we are, our question is, can this kind of catalytic complexes originate from prebiotic Earth, where there's more likely to be a pool of random sequences? To study this question, we did an in vitro selection, and this selection, as like Uli has introduced, was established in our lab in 2014 to select a triphosphorylation ribozyme. And we made a few modifications to the system so that we were able to select catalytic complexes from this selection. To start with, we generated a DNA template that contains 25 nucleotides long randomized region. And this region is relatively short because based on our previous study on the shortest triphosphorylation ribozyme, 25 nucleotides is likely to be too short to be able to catalyze the triphosphorylation reaction by itself. So use this short randomized region, we are trying to reducing the possibility of selecting a pool of short ribozyme that doesn't need any fragments. And then this DNA template was in vitro transcribed, and the hammerhead ribozyme at the 5' end will self-cleave during the transcription step, generating a 5' hydroxyl group. This RNA pool with the 5' hydroxyl group will then be incubated with trimethaphosphate, and then they will be challenged to triphosphorylate its own 5' end. Because we have a very short RNA pool, we added a second library contains 256 different DNA 20-mers to facilitate RNA to catalyze this reaction. This second library was chosen by Arvin, who was a PhD in our lab, to have no bias regarding the 4 nucleotide compositions and the self-structure tendency indicated by the predicted delta G values. So these 256 sequences can be regarded as a small representative of a random environment. An RNA pool will be able to recruit DNA cofactors that they can use to form catalytic complexes and catalyze the reaction. So after this incubation step, the fragments will be incubated with a biotinylated short oligo and a ligase, for example. Those RNA that has been triphosphorylated will be ligated to the biotinylated oligo, and then this long strand will be pulled down by streptic actin beads. In this way, we get rid of those inactive RNAs from the pool. And then these active sequences will be reverse transcribed into DNA, and then PCR amplified and also regenerate the template, and it's ready for the next round. In the first selection round, we started with an effective complexity of about 2 times 10 to the 14, which means the estimated number of different RNA sequences go through the first selection round is around 2 times 10 to the 14. And then these RNAs go through 6 rounds of selection. After each round of selection, we collect the selected pool, and we send all these different pools after different rounds of selection to high throughput sequencing, so that we can analyze how the sequences changes during the selection. And this is the high throughput sequencing analysis of the last 4 rounds of selection. We can see a lot of RNAs has been enriched over rounds. From the high throughput sequencing analysis, we have found more than 1,000 different RNA sequences that has at more than 500 reads. And this plot shows that they are enriched over rounds, and some are outcompeted over rounds. And then we also did in the last round of the selection, besides the original selection run with all 256 DNAs, we also did 18 sub-round selections. The 256 DNAs were divided or grouped into 16 groups, 16 sets, each contains 16 different DNA sequences. And we amplify, we do one sub-round of the RNA in the presence of different DNAs and a control group with no DNAs in presence. Then we calculate the sequence enrichment by normalizing the RNA, each RNA's abundance over the no DNA group, so that this plot showing that for each RNA, they are amplified, and then that indicates they are facilitated by different group of DNAs. So from this plot, we chose 45 RNAs that seems to have strongest effects caused by different DNA groups for biochemical analysis to identify the exact DNA sequences that is helping with different RNA molecules. And this is how we did the biochemical analysis to identify the DNA cofactors. For each RNA sequence, we first incubate them with 16 different DNA sets. And those RNAs, once they have been reacted, they will be ligated to radioactive labeled short oligonucleotide. And then we can run the product on the gel, and this upshifting band will tell us how much what percent of the RNA has been ligated. That means it has been triphosphorylated. So this is how we indicate the activity of the complexes. And then those DNA sets that has activate the RNA, we have found multiple sets of DNA, and in each of these sets, we test the RNA with the 16 DNAs individually. For example, this is a small group of DNA tested here, and we can find the individual DNA that is assembly with these specific RNA sequences. And we did the same to the other groups, and we found 4 different DNA cofactors that can activate this RNA fragment. And then we did the same type of experiment to all 45 RNAs. We found they have different preferences to the DNA cofactors. They use DNA from different groups, different sets. And then when we trying to cluster these RNAs based on their sequence similarity, the sequence similarity coincides with the DNA cofactor they use. These RNAs colored in the same color, they use the same group of DNA cofactors. And we found they require different number of DNAs for activity, or even without the help of DNA. And they use DNA from different DNA sequences. So this shows that there could be many different types of complexes formed, and they might form different secondary structures. So we chose 7 representatives from different clusters for more detailed characterization. And I will next showing 4 examples of the characterized complexes. And the secondary structures were is a model that's supported by our inline probing data. The first complex that formed by RNA R34 composed of 1 RNA and 3 different DNA fragments. And 2 of these DNAs are required at the same time for RNA activity. Without one of them, there's undetectable active triphosphorylation activity. And the third DNA cofactor can significantly increase the activity of the whole complex. We also test the kinetics under its optimum temperature at 30 degrees Celsius. And then, yeah, this is the fastest kinetics complex among the 7 complexes we have tested. And the second example is a RNA that use 2 molecules of same RNA and 1 molecule of DNA. And this is model is generated based on our titration of the DNA and also by inline probing data. And then this complex has no significant preference of temperature ranging from 10 to 40 degrees Celsius, and it has lower kinetics under 25 degrees Celsius. The third example, I took one example of 1 RNA, 1 DNA complex, and we have a few more examples of 1-to-1 complex, and they all form different secondary structures. And this one has optimum temperature at optimum temperature at 30 degrees. And then we also found 1 short triphosphorylation ribosome that doesn't need any help from DNA cofactor. And after truncation, this ribosome is 49 nucleotides long. And it also has no significant temperature preference, ranging from 10 to 40 degrees Celsius, and it has the slowest kinetics. So now we have found many different RNA-DNA complexes that can work individually. We are now interested in how they will behave when we pull them together, if the activity will be affected by the fragments by other fragments in the environment. So now we did, before we do the one-pot reaction, we extended and truncated those RNAs for a few nucleotides so that it can be separated on the gel and they can be simultaneously monitored in the one-pot reaction. And then we first pull the 7 RNA fragments together, and we incubate them with 256 DNAs. That was the original DNA library we used in the selection. And we found all of them active, and this is as expected because this is how we were able to select them. And then we are trying to study a more prebiotically relevant environment where there's more complicated sequences in the environment. And we change the DNA replace the DNA by the completely randomized DNA 20-mers. And we were surprised to see that 4 of these out of 6 RNAs that require DNAs that they are still active with the completely randomized DNA 20-mer. And the RNA that is active by itself, it also remains active in the more complicated environment. So this is interesting to us because it supports the idea that the fragments can be generated in a random environment. It can also recruit the DNA sequence it needs from a randomized environment. So to briefly summarize what we found from the selection, we found multiple different complexes formed by short oligonucleotides, and they can catalyze the reaction, and they evolved from completely randomized libraries. And then a more interesting result is that 4 out of 6 RNA that has been tested, they remain active in the presence of randomized 20-mers. Yeah, so this is what I have for this talk, and I would like to thank Ulrich Muller and all my lab members, especially Arvin, for generating the DNA library for the selection. And we thank NASA for funding and thank for the organizers.
Great, thank you, Shu. As a reminder, we'll hold questions till the end. All right, while he gets his slides ready, I'll introduce our next speaker, Alberto Vazquez-Salazar. He received his PhD at UNAM, Mexico, under Professor Antonio Lozano's supervision. And Alberto is currently a postdoctoral, a NASA postdoctoral fellow at UCLA and Professor Irene Chen's lab. And so he will now tell us about his work in vitro evolution.
Thank you very much. Thank you very much for that kind introduction. And thank you to the organizers for the opportunity to be here. I'm very, very happy to be part of the PCE 3 seminar series and to discuss with you some of the work we've done in Professor Irene Chen's lab at UCLA. Today, I'm going to be talking about chirality, the problem of chirality, and specifically chiral transfer using in vitro selected model ribozymes. And to start with this, the very basic things we have to remember is that amino acids and sugars exist as mirror images of each other. And these mirror images, these molecules, are known as enantiomers. For amino acids specifically, no matter how we adjust the orientation of these 4 substituents in the 3-dimensional space, it is impossible to superimpose them on the chiral carbon. And in extant biology, sugars and amino acids, and of course the polymers they form, exist exclusively or almost exclusively in one of these 2 possible enantiomers. So this means that life, as we know it, current life, consists basically of right-handed or V sugars and left-handed or L amino acids, and hence it's D nucleic acids and L proteins. And the advantages, the selective advantages of this homochirality, of having just one chiral species of these molecules, could be that they confer structural specificities and probably chemical efficiencies. And these selective advantages could drive natural selection towards a system with nearly complete or complete chiral asymmetry. So how do we study the emergence of homochirality? Well, we can consider three different stages for the emergence of homochirality. The first one being in the breaking of symmetry, in which an imbalance is introduced to a system with a racemic mixture of both enantiomers. The second step will be amplification of this imbalance, in which such imbalance increases until a stage of homochirality is reached. And finally, what we call chiral transfer, in which one enantiomeric species, in this case will be the RNA, interacts with another type of molecule, and this interaction passes or transfers the chiral imbalance to the second type of molecule, which in this case will be peptides, and then will be proteins. And obviously this chiral transfer at the end is going to break the symmetry of this second type of molecules. And as Uli mentioned before, in the context of the origin and early evolution of life, it is hypothesized that there was a very early period of evolution in which RNA played a very important role as both the genetic carrier and the catalyst. This stage in early evolution of life is what we know as the RNA world. And it is also hypothesized that during this RNA world, the origin and the early evolution of the protein translation system started. So that will mean that the interaction of the RNA with the protopeptides and the first proteins could have also been important for the chiral transfer of from an RNA molecule to a peptide. This means that, since coded proteins are theorized to have developed from the RNA world, then the homochirality observed in L amino acids might be the result of a chiral transfer from a homochiral DNA. So DNA gives rise to L amino acids and L proteins. And how do we study this, or what do we do about this? So it has been proposed that this RNA, the RNA, may have an inherent affinity for reaction with L amino acids or even their precursors. And this will mean that the RNA world deterministically and inevitably will lead to an L protein world. But is this actually true? Well, so far there's some evidence that supports this bias of reaction between the RNA towards L amino acids. But in reality, the proposal that the dRNA world intrinsically leads to a L peptide world needs more experiments and further testing, especially what will happen if we use an alternative amino acylation structure in what we are calling like a type of alternative RNA world. Will dRNA still favor the reaction with L amino acids? So in the lab, as Uli mentioned also, in previous work, we have identified and discovered through in vitro evolution experiments ribozymes with the activity of self-amino acylation. This means that these ribozymes can interact with the substrate, which in this case is a biotinylated 5,4-H oxazolone, an analog for the amino acids that we're using, in this case, an analog for phenylalanine. And these ribozymes can interact with the substrate and actually incorporate the substrate into their structure. And they do it by an ester bond with an internal, which is important, an internal 2' hydroxyl group. And in this case, in this ribozyme, one of the ribozymes I'm just exemplifying here, we know where the reactive nucleotide is, which in this case is this G65. These ribozymes come from in vitro selection experiments with a random region of RNA molecules with a random region of 21 nucleotides. And in the previous work, in the previous in vitro experiment, 3 different sequence motifs were identified. So what we use for these experiments that I'm going to describe next are ribozymes that are representative members of these 3 different sequence motifs and sequence families. And one thing that is very important for this experiment is the nature of the substrate. As I mentioned before, is a biophenyl phenylalanine oxazolone. This is the oxazolone part, and this red asterisk here is showing the chiral center of the amino acid fraction for our substrate. So what this means is that when it's in solution, there's an racemic fraction of both the phenylalanine and L-phenylalanine. So the ribozyme and the ribozymes are actually interacting with both species, enantiomeric species of the substrate, both the D and the L. So the system turned out to be a very nice system to test the hypothesis of the hypothesis of these V ribozymes interacting exclusively with L amino acids. So in order to test that, we first incubate our ribozymes, our RNA sequences, with the substrate, which again is in a racemic mixture in solution. And once we have the amino acylated fraction of the ribozymes, that is going to look like this, we are going to be able to selectively hydrolyze the ester bond, and we are going to be able to separate the RNA from the biotin phenylalanine via exclusion chromatography. And after that, we can cleave the amide bond and eliminate or separate the phenylalanine and the biotin. And what we're actually doing is isolating the 2 different enantiomeric species, both L and D, and then we can analyze that mixture of both L and D enantiomers with a HPLC on a chiral column, and then we can just analyze the peaks by mass spectrometry and determine the ratio of each enantiomeric species, of each L or D molecules. And with this, we can test the hypothesis if our ribozymes, which are again our D ribozymes, prefer the L enantiomeric species or the D enantiomeric species. So this is just to show that all the ribozymes that we used, this is a representative of the ribozymes we used, 15 ribozymes. This is just to show that the ribozymes do have self-amino acylating activity. This is a streptavidin shift assay in which the shift in the banding pattern here is indicating activity. So every time we have this band, this upper band indicates amino acylated RNA. And then after the selective hydrolysis of the amino acyl ester bond, what we find is that we don't see the streptavidin bound amino acylated fraction of the RNA, meaning that we're actually getting rid of the streptavidin and of the biotin later. So once we have the isolated D-phenylalanine and D-phenylalanine in solution, we can go to the HPLC and on the chiral column, and we can verify the identities of every single enantiomeric molecule. So what I'm showing here is just how we do the standards. We use tryptophan, and phenylalanine, the amino acids, like in equimolar solution, 10 micromolar, to run on the experiment and see how the standards work. And then we run these standards with the tryptophan with the product of activity of a very well-studied ribosome in the lab, which is named S-1A.1-A. This molecule we know is extremely active with the substrate. So we incubated this ribosome with our substrate at this concentration just to see if we could actually see a difference, like a start point for the experiments. And as you can see, once we analyze the peaks via mass spec and identify the identity of the L and D enantiomers, we can start to see that there's a difference between the preference of this ribosome in a specific with each one of the enantiomers. So for our experiments, we use 15 different ribozymes. Again, these ribozymes are representatives of the 3 different sequence motifs that were found in the previous in vitro selection experiment. And we did a series of reactions with the substrates at different at different concentrations of that substrate. And we measure the amino acylated fraction versus the non-amino acylated fraction of the ribozymes, and then we identified which enantiomer was being bound to the ribosome. And as we can see, we have a strong preference for most of the ribozymes, of the 15 ribozymes, to be binding to the V enantiomer, like the case of members that start with the number 1. But for ribozymes that are members of another ribozyme family, those that start with the number 2 here, we observe something very interesting, and that's that the preference for the enantiomeric species of phenylalanine is not really the D amino acid, but rather the L amino acid. And we were able to determine the catalytic rates for all of these 15 ribozymes, and to estimate with those catalytic rates, the binding affinity for the D amino acid or the L amino acid, we were able to calculate many other things, including the enantioselectivity index, which is defined as the log2 of this ratio of the catalytic the catalytic constants for the binding of D or L enantiomer. So what we can observe is that when we analyze the enantioselectivity index, a negative value of this number, which comes from this log of the ratio of the catalytic constants, again, a negative value indicates a preference for the L isomer, but a positive value indicates a preference for the D isomer or the D enantiomer. And what we can see here very clearly is that 6 of the 15 ribozymes are actually showing preference for the D isomer of the phenylalanine analog. And that is very, very interesting, because that goes exactly against this generalized hypothesis of DNA having this intrinsic preference for L amino acids. So we just basically, after we had this data, we played with the enantioselectivity index. So what I'm showing in this panel here, the left panel, is the distribution of the indices that shows that there's actually a range of values, meaning that every single ribozyme has a different preference for a specific type of enantiomer. But as we can see here, there's some ribozymes that have a stronger preference for the L amino acid, whereas there's other ribozymes that have a stronger preference for the D the D enantiomer. And there's some ribozymes that have just a slight preference for the L enantiomer, but still, after doing the math and the calculation, there's still a preference for the L enantiomer. So that was for us a very, very cool and neat result, because again, we are showing that there is really no intrinsic preference from the D RNA towards an L amino acid. But of course, analyzing the structure of our substrate, it's very complex chemically. And of course, it includes a biotin group that is needed or was needed for the in vitro selection experiment to pull out the sequences that actually reacted. And this biotin group itself has chiral centers. So one of the questions that we asked ourselves was, is it possible that what we are measuring is an artifact and is not really the chiral center of the amino acid, but rather a chiral center in the biotin in the substrate? And for this, we did this experiment, which is, I think, one of my favorite experiments of this whole project, in which we choose, again, this ribozyme that we know is very reactive, and the D ribozyme, like the normal RNA molecule, reacts with the L amino acid. Yeah. So what the hypothesis is, if our ribozyme is actually interacting with the chiral center of the substrate, if we switch the chirality of the ribozyme, and now we have a L RNA, which they call here, for instance, the evil system, but if we have an L RNA and the RNA is actually looking or interacting with the chiral center of our amino acid analog, then when we make those two molecules, the L RNA, the evil system, and the substrate interact, the preference is going to also switch if it's interacting with the chiral center of the substrate rather than with other chiral centers in the substrate. And that's exactly what happens when we have our L RNA. The preference for the enantiomeric species of the amino acid also switches. And this is corroborating that the ribozyme, the chiral ribozyme, is actually interacting with the chiral center in the amino acid analog. So that means that further contributes to the idea that there is really no such thing as inherent reactivity between the DNA and L amino acids. It's more dependent on the structure that is being used for the catalytic activity and the sequence of these structures. So as conclusions for these whole projects and set of experiments, we have that the ribozymes that were analyzed, specifically those of motif 1, were found to be selective for the D-phenylalanine enantiomer, which was expected over the were found to be selected for the D-phenylalanine over the L substrate. And this finding contradicts the hypothesis that D RNA has an intrinsic general preference for L amino acid. Instead, we conclude that the chiral preference appears to depend on the specifics of the system, and either outcome, the D protein or L protein, or D amino acid and L amino acid, would have been actually possible. And this depended on the structures used for amino acylation in the RNA world. And this also means that just small imbalances could have been amplified by natural selection or other chemical or physical mechanisms, meaning that if in the system, in the prebiotic system, there was just a very subtle imbalance, and that imbalance made a RNA molecule to interact with a certain enantiomer rather than the other, that interaction could have been amplified by natural selection. So one of the final conclusions of this work that may sound interesting to some people is that chance is extremely important in the history of life, and obviously in evolution, and in this case in ribosome discovery, exploration and discovery. So the ultimate emergence of an L versus a D protein world might have been as random as a coin flip. And with that, I want to thank and acknowledge the Chen Lab, Professor Irene Chen, my PI, Dr. Josh Kenchel, which is a co-first author with me of this work, and all our collaborators, and of course the NASA postdoctoral program in astrobiology. And thank you very much.
Okay, thank you, Alberto. So now we should have time for questions. There are two ways we can organize these questions. First is you can type your question in the chat, and we'll go through each one. Alternatively, you can virtually raise your hand through Zoom, and you can ask your question. While people are formulating their questions, I think you might have actually addressed Donna Blackman's question during your talk. Unfortunately, she had to go, but she asked about the fact that the sorry. Yeah, sorry, I couldn't find this. That biotin already has the chiral center. Could this the differences in reactivity between those? I think you might have addressed her question.
Yeah. So what we did was to so there was two ways, well, probably more than two ways to address this. One would be to try to get rid of biotin and keep an actual amino acid mixture or amino acid analog mixture in the system that reacts with the ribozymes. But biotin is actually very important for the reaction because the original in vitro selection was done with biotinyl oxazolones and so on. So the thing we tried to solve that was, instead of changing the substrate, was changing the chirality of the ribozymes. And when we switched the chirality of the ribozymes, we found that the preference for the enantiomeric species of phenylalanine is also switched. So that was a very interesting result for us because, again, that means that even though biotin is important because of the original in vitro selection experiment, the catalytic nucleotide in the ribozyme and the structure of the ribozyme is actually interacting with the chiral center of the analog of the amino acid, or the chiral center is what's really driving the amino acylation to the ribosome, meaning that biotin doesn't really play a role in which enantiomer and amino acid it's binding to the ribosome. So yeah, it's definitely something very important to keep in mind, that biotin has by itself chiral centers, but I think that we solved that issue with that very nice experiment, switching the chirality of the RNA.
Very interesting, very thorough. All right, do we have any other questions? Let's see for raising hands. All right. For Shu, I can ask a question. So I might have missed part of this, but when the DNAs were incubated in your selection experiments, how did you go back and assign the structures of the DNA RNA complexes?
So we first need to identify which DNA sequence was used by a certain RNA sequence, and that is we just simply test all DNA in groups. So what I showed that two-step biochemical assays, we first identify which DNA sequence from which group the DNA sequence was active, and then we test all 16 DNA from that active group, and then we can get the specific DNA sequence that is reacting with one RNA. And then to test the structures, we do inline probing. We just incubate the RNA with the DNA in the buffer, and we can read by the inline probing data, we propose a possible secondary structure.
Okay, so from AC for Alberto, sorry, a bit confused. I thought that you showed the LRNA switched the amino acid substrate specificity. So doesn't that show the RNA chirality selects for the amino acid chirality?
Yeah, I apologize if I said something that's not it, but that is exactly what happened. RNA chirality selects for the amino acid chirality. So the amino acid selects for either L or D. And when we switch the chirality of the RNA, we also switch the selectivity for the chirality of the amino acid. So yeah, what you said at the end is exactly what's happening.
Great. We have question. Sure, Jack. I was trying to unmute you. I'm sorry.
Hello everyone, I am Shisha from India, and I want to say a few things that how can we hypothesize or what is the experimental proof that only the RNA might have been need affinity for the reaction of L amino acids? Means is there any experimental evidence found yet?
Yes, thank you very much for the question. There is several experiments and published papers that try to understand this issue, and they also have RNA system. The one I remember right now is an analog for the tRNA. They use a mini helix. I don't know if you're familiar with like the mini helix model for tRNA evolution, but they use the mini helix and react that mini helix with either L or the enantiomers of amino acids, and they constantly show that the mini helix prefers the L amino acid. So that experiment and other, around the same line of RNA being reacted with a racemic mixture of amino acids led to results showing that RNA reacts with L amino acid. So at one point, it was kind of, I don't know if established will be the right word to use, but it was proposed that, yeah, RNA had this intrinsic chemical preference for the L amino acid, and that's why in the early stages of evolution, once we have an RNA world, and once the RNA of that RNA world, which is a D RNA, started to interact with amino acids and then peptides and then proteins, those were L amino acids and L peptides and L proteins. So, and then I think probably Professor Donna Blackman could correct me here. but I think she left. I think after that series of experiments, people just kind of stopped doing more exploration on the topic. So it was a kind of understatement that RNA just reacts with L amino acids and hence L peptides and L proteins. But yeah.
Okay, so a follow-up on that. AC asks, so why the coin flip conclusion?
Yeah, it's because randomness has a huge role in the evolution of life and in evolution itself. And we chose the coin flip analogy because if at the very early stages of evolution, we have RNA interacting, let's say, with a racemic, like a truly racemic solution of enantiomers, that means 50L and 50D, that's a 50% chance of actually reacting with either one of those. But if something happened in the system, a small invariance happens in the system, and the RNA reacts with one of the two enantiomers, and then that interaction with just the one enantiomer amplifies the reaction of RNA with that. That could be like the starting material for evolution to just work with, let's say, V RNA and L proteins. And that, because of the like how random the imbalance, small imbalance was introduced, or even how random it is to just go with an RNA interacting with L or an RNA interacting with D, that's how we made the analogy with a coin flip. Because as we understand it and as we conclude, could have been as random as just choosing from a 50% RNA D or a 50% RNA L. It's just to emphasize that random, like true randomness, true random events played a huge role in evolution, rather than like an actual selective advantage for L or D, or a chemical intrinsic preference for L or D. And yeah, the prebiotic, not the RNA pool, but the amino acid pool should have been racemic. And more than our hypothesis, our assumption at this point is that RNA in the RNA world was a D RNA world. And of course, how we got from a racemic mixture of sugars to an RNA world is a whole different discussions. But I know that Professor Donna Blackman has papers where she shows that again, a small imbalance in the system can lead to the formation of RNA from a racemic mixtures of sugars and precursors. So there's a lot of questions involved in this, but at least for our work, we have the assumption that there was an RNA world and a racemic mixture of L and D amino acids.
Alberto, I have to say, I really love the general conclusion that your research has reached, because I think in this study of the origin of life, it's implicitly or sometimes even explicitly treated as if some of the things that ended up in life today are deterministic. And I really like the general conclusion that no, that's not necessarily the case. It could have been otherwise. It just had to choose something, and that's what we ended up with.
Yeah, definitely. And again, I feel like as a biologist, I was taught that, yeah, randomness and those true random events play a huge role for the evolution and, of course, the early evolution of life. So, yeah, when we still don't have these machineries to actually select and like do all the whole process, back then in the very early stages, true randomness and true random events must have been extremely important. And yeah, just that, random. And
Yeah, I completely agree.
Okay. I think I actually have another question for Chu. Back to your RNA DNA complexes. And you might have mentioned this, but did you try to replace those DNA fragments with RNA to make it like an all RNA complex? And did they function the same or perhaps function better as an all RNA complex?
Yeah, thank you. That's a very good question, and we haven't done that yet, but that was in our plan. That is when we designed the selection, we are thinking about whether to use RNA or DNA, and DNA is easier for my selection, and but we are thinking, yeah, it's possible to change the DNA with the same sequence but in RNA form, and that is what we are interesting to follow up on.
I was interested in the temperature profiles that you showed. And, there's a general consensus that at least in the duplex form that, RNA duplexes might be a little bit more stable. So I was thinking, maybe you might get some increased activity just owing from the fact that it seems that all RNA, especially ribozymes, always tend to misfold. And
Yeah,
so that would be interesting to see.
yeah, thank you.
Okay, and then we could probably ask this one last question. AC from AC asks, are you analyzing other amino acids besides phenylalanine? Would glycine have the same results?
so there's several small answers for that question. So first of all, we used phenylalanine because there was a lot of standardized techniques and methods already published on how to analyze chirality of phenylalanine. And also, phenylalanine is one of the amino acids we actually can synthesize in the lab and work with them. Others include tryptophan, tyrosine, valine, isoleucine, methionine, and leucine. And we have those amino acids specifically because they are like we can synthesize them in the lab, not because we're unable, just because the chemistry doesn't really work for other side chains to synthesize the hexavalent form of them. And so that's why we use phenylalanine. Currently, we're not analyzing other amino acids of our catalog for the chirality of the ribosomes. We think that this result could be general to the other amino acids. And glycine, even if we could synthesize a glycine analog that reacts with our ribosome, glycine has the side chain of glycine is a hydrogen. So it actually has two hydrogen substituents, making it not chiral. So glycine is the exception to all the other amino acids in which it's got no chiral center. So there's really nothing to select for in glycine in terms of a chiral center.
Okay, well, think there are no more questions. All right, thank you for the wonderful talks. Great, yeah, okay. Yeah, thank you for the wonderful talks. Appreciate these. Okay. See you all at our next PC3 series.
Yeah, thank you so much, Beito and Chu, and thank you, Yuli. Great presentation.
Thank you. Thank you.
Yeah,
Thank you all.
thank you. Thank you.
Yeah, see you all next time.